I'm Dr. Sue Jarvie, and I'm the head of the Hawaii Island Rat Lungworm Working Group, housed here in the Daniel K. Inouye College of Pharmacy at UH Hilo. Rat lungworm disease is also called neuroangiostrongyliasis, which is a mouthful, and so we'll just call it rat lungworm disease for now. And it's caused by a worm, a nematode, and I just want to talk about the life cycle for just a few minutes. And so it involves rats, that's why it's called rat lungworm disease, and it reproduces in the rat. And the rat, in the rat feces is the first stage larva. The first stage larvae then have to develop in an intermediate host, and that intermediate host is a slug or a snail. And in the slug or the snail, it develops to the third stage larva. It goes from first stage to second stage to third stage. And it's that third stage larva that's infective back to rats. And so rats love to eat slugs and snails. And so the rat will eat the infected slug and will become infected. When humans get rat lungworm disease, it's thought that most people get rat lungworm disease because these infected larvae are in water, for example, or in beverages, or on unwashed produce, and it's ingested. And when people get rat lungworm disease, they can get very, very sick. Rat lungworm disease isn't unique here to Hawaii, but it's present in over 30 countries worldwide. It's considered a global emerging infectious disease. Hawaii, especially East Hawaii Island, has a distinction of having the highest number of rat lungworm cases in humans in the United States. Even though there are cases throughout the state, and also on the mainland now, East Hawaii has the highest number of rat lungworm cases. And so one of the questions that we're asking is why is this? Why do so many cases originate from East Hawaii Island? And so to address this, we're doing a number of research projects. And those projects are all based on questions. One of the questions we ask is how infected are the slugs and snails here in Hawaii? We've done some testing in rats. We collected over 500 rats and looked at them. I can't show you that right now because we're not going to dissect a rat, but we looked at them and counted the number of worms that we could find in those rats. And 94% of those rats were infected with rat lungworm parasites. So that's a really high infection rate. We also want to learn a little bit more about transmission of the parasite. We're asking the question, can the parasite be transmitted in water? And we've had cases here in Hawaii, human cases, where people have gotten infected through drinking a beverage where a slug had climbed in it and the larvae have escaped. And so we want to find out the potential for transmission through water. Especially here in East Hawaii, we have a lot of people on water catchment. They don't have municipal water. And so this is another potential route of transmission. Another question we're asking is how effective or how sensitive are these larvae to anti-parasitic drugs? And so we're running a study right now to test that. In order to do all these studies where we have live larvae and we want to treat them in one way or another, we need to know whether that larva is dead. We can tell when it's alive, and we'll show you pictures of live larvae because the larvae are wiggling under the microscope. And so we're developing an assay that we'll be able to use to actually establish that after we treat them with a vegetable wash or heat or freezing temperatures for a certain amount of time, that those larvae are actually dead and therefore non-infective. Right now, if you think you had rat lungworm disease and you went to the hospital, they would want to do a spinal tap to get some of your cerebral spinal fluid to be able to use it to determine if there's parasite DNA. They do a molecular test on that fluid. And a lot of people are a little hesitant to get a spinal tap. And so one of the other projects we're working on is developing a blood-based diagnostic that could be used for detecting antibodies against rat lungworm disease. It could be used in humans, and it could be used in animals because our pets, our dogs and horses especially, also get rat lungworm disease. So we'd love to bring all of you into the lab so you could see what we're doing, but that's just not possible. So we decided to make this video to bring the lab and the research projects we're working on to you. And so today you'll learn how to isolate larvae from a slug and how we extract the DNA from that larva, and then we can determine how infected that slug is. And you'll also learn a little bit about how we have developed a blood-based diagnostic. And we don't recommend that people handle slugs or snails with their hands. You don't know if they're infected or not. So we in the lab use a lot of protective equipment when we're working with live larvae because they are infective. We wear a mask to cover our mouth and nose so nothing splashes. We wear goggles and we wear gloves, lab coats, okay, long pants and solid shoes. And that way we're fairly well protected.